Assalamu alaikum. This is Dr. Hina. Today we are going to discuss an important topic, which is digestion and absorption. In this presentation, we will cover digestion of carbohydrates, proteins, and lipid, the absorption mechanism in small intestine, the role of intrinsic factor in vitamin B12 absorption, key digestive enzyme, and malabsorption syndromes. The overview of the digestive system, as we have already studied, that digestion is the process of breaking the food into smaller particles so that they could be easily absorbed. Absorption again is followed by digestion, and in it the moving nutrients from the GI tract into the bloodstreams takes place, whereas the transport is the absorbed nutrients being transported throughout the body via your circulatory system and lymph system. Coming on, the last process is elimination, and elimination is basically of the undigested or unabsorbed food through the feces. Here you can see in the diagram that a person first ingests food through the mouth through the help of the teeth and chewing, which is process is called as mastication. It tears and shreds the food into smaller particles, then it follows into the pharynx, and to prevent the bolus from entering into the trachea, the epiglottis closes, right? Because pharynx is a common pathway for your air, respiration, and your food. Now, entering from the pharynx, it passes into the esophagus, and once it passes close to the stomach, there is a lower esophageal sphincter which relaxes after swallowing to allow the bolus to enter the stomach. After entering the bolus into the stomach, the lower esophageal sphincter then contracts and prevent the stomach from returning the contents into the esophagus. And if, for example, if this lower esophageal sphincter does not contract, the food can enter into the esophagus, and a very common symptom which is found in patient is heartburn and your indigestion, or you can also call as GERD, which is a gastroesophageal reflux syndrome. Now, as the food from the esophagus enters into your stomach, here the stomach mixes and churns the food in with the acid, the enzymes, and the gastric fluid which is present, and this liquid which finally forms after the mixing of the food is called as chyme. Now, obviously the stomach then continues into your small intestine, and the small intestine secretes further digestive enzymes to absorb the nutrients, whereas your large intestine then after it passes through the small intestine, the bolus of food, then it enters into the large intestine, and it reabsorbs the salt and water which are needed by the body, and the unwanted product is being wasted or is then passed on to the rectum to be. Now, like the parietal cells are responsible for the secretion of HCl, whereas your chief cells are responsible for the secretion of pepsinogen, and from the pepsinogen it activates into pepsin, which further causes the digestion of proteins. From the stomach, the chyme enters into the small intestine where it is further absorbed and digested, and here the small intestine is divided into the duodenum, jejunum, and ileum. Now, once the food enters from the stomach into the duodenum, it is through the pyloric sphincter, and till the ileocecal valve the food travels. Now, it takes around 3 to 10 hours for the food to traverse. Now, small intestine is further specialized for the absorption of food with its multifolded arrangement, which provides a greater surface area for the nutrients to be absorbed. Now, once they are absorbed, they enter. How they are absorbed? Through the finger-like structures of the villi. The blood and lymph vessels pick up these nutrients through the brush borders, and then it is transported throughout the body. Now, these brush borders it consist of microvilli, which are basically the brush border, and these microvilli are responsible for the further increment of the surface area. अच्छा, surface area increase करने का क्या फ़ायदा हो रहा है? Because why we need a much larger surface area? The body could be in a specific arrangement, right? अगर हमारे पास बहुत बड़ी intestine होगी, so it will not be easy for the body to move around. इसीलिए उसी intestine के अंदर further partition होके villi हैं, and then those are then further divided into the brush border, which are the microvilli, and this increases the surface area so that there is maximum absorption of food. Okay, traveling from this the whole digestive system. Now see, there is the ileocecal valve, then you have the ascending colon, the transverse colon, and the sigmoid colon. ठीक है, sigmoid colon से पहले आपके पास descending colon होता है, and from the sigmoid colon it enters into the rectum, and from the rectum then finally it is excreted out, stored there in the rectum. The undigested unwanted food is stored there, and then it is further excreted out. The large intestine is a site for the absorption of water, sodium, potassium chloride, all the electrolytes, the important electrolytes which are present in the large intestine, which is basically the absorption process taking place. Whereas your bacteria in the large intestine produces vitamin K, thiamine, riboflavin, biotin, and vitamin B12, and only biotin and vitamin K can be absorbed in the large intestine. See, so there is only the absorption of two vitamins, which is biotin and vitamin K. Can you explain me why vitamin K is very important? Okay, vitamin K is important for your clotting mechanism, very important for the activation of your clotting factors. ठीक है, so इसीलिए when a child is born, usually they are given an injection of vitamin K, and that is considered to be helping in clotting of the blood in the children or in the infant who is born. Okay. Now, in the large intestine, there is 1 liter of fluid material which gradually is reduced to how much? To 200 grams of brown fecal material. So, imagine 1000 milligram or 1000 ml being converted into 200 grams. Okay, and this brown color is due to the unabsorbed iron. Okay, so the brown color is basically due to the unabsorbed iron with a yellowish orange substance called as bilirubin. Now, greater the iron content, the darker the feces. Elimination. Finally, the elimination takes place, which is to the rectum, which are having warehouses waste before the release to the anus, and then the anal canal relaxes and releases the waste. Fine. Now, the functions of each region are different in the digestion process. Like in the mouth, the starch hydrolysis takes place, and this is catalyzed by amylase, which is secreted by your salivary glands. The fat hydrolysis takes place with the lingual lipase, which is secreted by the tongue, and absorption of small amounts of vitamin C and a variety of non-nutrients. Okay. Now, here you can see we have mentioned two major substances for digestion, which is starch and your fat hydrolysis. Hydrolysis means breaking down. Starch is a complex carbohydrate, okay, which is polysaccharide, whereas fat is further catalyzed into small fatty acids. Then you have stomach, which is responsible for the denaturation of dietary proteins and release of vitamin B12, iron and other materials from the protein binding, for which gastric acid is important. See why there is gastric acid, why there is HCl in the stomach? It is for the release of vitamin B12, for the protein binding of the minerals, okay. And so the protein hydrolysis is catalyzed by pepsin. The enzyme pepsin is present. For the fat hydrolysis, the enzyme lipase is present, and secretion of intrinsic factor required for absorption of vitamin B12. Now, here we have mentioned intrinsic factor. Intrinsic factor is secreted in the stomach and which is responsible for the absorption of vitamin B12. Okay, if there is a deficiency of an intrinsic factor, what will happen? We have already studied this in the blood module. Overview of small intestine. The small intestine, duodenum and ileum is responsible for further hydrolysis of the starch by amylase secreted by the pancreas. So here this amylase is called as pancreatic amylase. Hydrolysis of disaccharides within the brush border of the intestinal mucosa. The fat hydrolysis is catalyzed by lipases, which is again secreted by the pancreas. So all the juices which are coming into the small intestine are majorly from the pancreas, okay. So they are basically coming from the pancreas. The protein hydrolysis is catalyzed by variety of exo and endopeptidases, okay, which is secreted by pancreas again and the small intestinal mucosa. So here the small intestinal mucosa is responsible. Hydrolysis of di and tripeptides. See, they are exo and endopeptidases, and there are di and tripeptides which are within the brush border of the intestinal mucosa, and the absorption of the products of digestion takes place. Absorption of water, failure of water absorption as in diarrhea can lead to serious dehydration. Coming to your large intestine, the bacterial metabolism of undigested carbohydrate shed intestinal mucosal cells, okay. So the undigested carbohydrates metabolism and shed intestinal mucosal cells, absorption of some of the products of bacterial metabolism, and absorption of water. Major is water, and your electrolytes are being absorbed in the large intestine. In the rectum, there is storage of undigested gut contents prior to evacuation as feces. Now, when we have digestion and absorption of carbon dioxide, we should know there are two types of sugars. Okay, sugar is similar to your carbohydrates, right? So both monosaccharides and disaccharides are divided into two groups, which are sugars contained within the plant cell wall. In foods, these are called as intrinsic sugars, where the sugars that are in the free solution in foods and therefore provide a substrate for oral bacteria, leading to formation of dental plaque and caries, and these are called as extrinsic sugar. Now these are all the classification you can see here. Polysaccharides, which are starch, oligosaccharide, which are dextrins, sugar, alcohols, then your sugar monosaccharides, disaccharides, which concludes your intrinsic sugars and your extrinsic sugars in free solution. And these extrinsic sugars are further divided into lactose in milk and non-milk extrinsic sugars, right? Now the starch is one of the major polysaccharide which we consume in the form of rice, right? And in starch in potato is very common, and then there are non-starch polysaccharides. Now digestion of carbohydrate takes place when the food is chewed and mixed with saliva, and which then contains the digestive enzyme which is called a ptyalin or an alpha amylase, which is secreted by the parotid glands. ठीक है, parotid glands obviously secretes the saliva. In the saliva it consists of ptyalin or alpha amylase. This enzyme hydrolyzes starch into disaccharide, maltose and other small polymers of glucose that contains 3 to 9 glucose molecules. Fine, so basically this enzyme is hydrolyzing the disaccharides, maltose and other smaller polymers of glucose, and they consist of 3 to 9 glucose molecules. Only around 5% of the starch is digested, because in the mouth the food remains for a very small time. Okay? So when it is for a very shorter time, the food, it cannot be completely digested. Now, at stomach, activity of salivary amylase is blocked by the acid of the gastric secretion. Of course, from the mouth it enters into the pharynx, and pharynx into the esophagus, and then this food, when enters into your stomach, the action of salivary amylase finishes, and 30 to 40% of the starches will have been hydrolyzed, mainly to form maltose. Now, the digestion of enzymes with the pancreatic secretions takes place or contains large quantity of alpha amylase within 15 to 30 minutes after chyme empties from the stomach. So, you are not having digestion of the carbohydrates in your stomach. You basically have in the mouth, then that digestion stops, or it pauses for a few time in the stomach, and after that it continues in the duodenum or in the small intestine, and through the large quantities of alpha amylase from the stomach into the duodenum and mixes with pancreatic juice, virtually all the carbohydrates will be digested in the small intestine, right? So they were not completely digested in the mouth or through the saliva, but they will be completely digested in the small intestine. Carbohydrates are almost totally converted into maltose or other very small glucose polymers before passing beyond the duodenum. Or the enterocytes lining the villi of the small intestine contains 4 enzymes, which are lactase, sucrase, maltase and alpha dextrinase, which split the disaccharides, the lactose, sucrose and maltose into their constituent monosaccharides. So the major monosaccharide produced in the body, in our body, is basically glucose. Okay, so glucose is a monosaccharide. These enzymes are located in the enterocytes covering the intestinal microvilli, the brush border. So we studied that the small intestine consists of alpha amylase, which comes from the pancreas, and it is producing its own enzymes which are coming from the enterocytes of the villi, and these are lactase, sucrase, maltase and alpha dextrinase. So you have 2 major enzymes which are coming from the small intestine, and one from your saliva, which is being produced by your parotid gland. Now maltase catalyzes the hydrolysis of maltose to 2 molecules of glucose. The sucrose or isomaltase is a bifunctional enzyme that catalyzes or hydrolysis of sucrose to glucose and fructose, and of isomaltose to two molecules of glucose. So maltase producing two molecules of glucose, the sucrose producing two molecules of glucose, then lactase then hydrolyzes into glucose and galactose, and thrallase catalyzes the hydrolysis of threalase of two molecules of glucose. So these are all the enzymes which are acting onto the different types of sugars and finally converted into glucose, which is easily absorbable and easily transferred into. Now this lactose splits into a molecules of lactulose and a molecule of glucose, where sucrose split into the molecule of fructose and molecule of glucose. So here they have shown us like how the starches enters through the saliva, then the pancreatic amylase digested, then the maltose and 3 to 9 glucose polymers further into the glucose, your lactose then forming glucose, and your sucrose in the intestine from fructose, and finally it is also connected together. So this is the diagram given below, the flow chart given below. You can easily understand from here the digestion of carbohydrates. Now coming to the digestion of proteins, the endopeptidases cleave proteins by hydrolyzing peptide bonds between specific amino acids throughout the molecules, whereas the exopeptidases remove the amino acid one at a time from either the amino or the carboxyl end of the molecule, again by the hydrolysis of peptide bond. Now the first enzyme to act on dietary proteins are the endopeptidases, which is the pepsin present in the gastric juice, right? We have studied that starch is being digested, basically carbohydrates are digested in the saliva, and when the bolus of food enters into the stomach, it then mixes with the gastric juices, which consist of pepsin, which is the endopeptides, where trypsin, chymotrypsin and elastase is secreted by the pancreas into the small intestine. The result of combined action of endopeptidases is the large protein molecules are broken down into smaller polypeptides, and with a large number of amino and carboxyl terminal for exopeptidases to act. ठीक है, पहले endopeptidases divide into or breaks it down into smaller peptides, and then these smaller peptides with polypeptides which are being formed are then acted upon by the exopeptidase. Now the exopeptidase consist of carboxypeptidases and aminopeptidases. The carboxy and peptidases are secreted in the pancreatic juice, which release amino acids from the free carboxyl terminal of peptides, whereas aminopeptidases are secreted by the intestinal mucosal cell, which releases amino acids from the amino terminal of the peptide. So carboxy can easily, the name itself indicates that there is a free carboxyl terminal of peptide, whereas aminopeptidases tell us that it gets the release the amino terminal of the peptide. Now pepsin is the important peptic enzyme of the stomach, which actually is active in your pH of around 2 to 3, which is a very acidic pH, whereas pepsin can digest collagen fiber. Collagen, from where is this coming? From the meat you are eating. Okay, the protein comes from the meat we eat, and of course the collagen fiber is present in the meat, and only 10 to 20% of the total protein digestion is to convert the protein to proteoses, peptones and few polypeptides. Now most of the protein digestion occur in the upper small intestine, which is your duodenum and jejunum. Okay, from the stomach finally it enters into the duodenum. Duodenum में digestion start हो जाएगी, which is through the enzyme which are trypsin, chymotrypsin, carboxy polypeptides and proelastases. Now these splits the protein molecules into small polypeptides, and so the carboxy polypeptides leaves them to the carboxyl ends, whereas the proelastasis converted into elastase and they digest the elastin fibers that particularly hold meats together. ठीक है, सो अब आपको यहाँ पे क्लियर हो गया होगा कि हाउ द प्रोटीन इज़ बेसिकली द मीट वी कंज्यूम, and that protein is then further divided into or broken down into polypeptides through different enzymes. Only a small percentage of proteins are digested all the way, ठीक है, to the consistent amino acid by the pancreatics. Most remains in the dipeptides or the enterocytes, which are present at the brush border, consist of hundreds of microvilli, and these are increasing the surface area. So these are multiple peptidases that are secreted that protrude through the membranes to the exterior. And the two types of peptidase enzymes are especially important are amino polypeptidase and several dipeptidases, which inside the cytosol of enterocytes are multiple other peptidases. So this slide clearly tells us that the brush border, the microvilli of the small intestine, again consist of peptidases which are responsible for the digestion proteins. Finally, coming on your digestion of fat. Fat is again a small amount of triglycerides is digested in the stomach to the lingual lipases, as we have discussed, and then the first step in fat digestion is physically to break the fat globules into a smaller particles to increase the surface area. And this type of digestion or the process, the first step is called as emulsification of fat, and this emulsification basically occurs in the duodenum through the bile which is coming from the bile duct from your liver. Now the bile salts and the phospholipid lecithin are extremely important for emulsification, breaking down of fat into smaller particles or making it increasing the surface area for the lipase enzyme to easily digest the fat globules. The process is through the digestion of triglycerides is pancreatic lipase present in enormous quantities of pancreatic juice. In addition, the enterocytes of small intestine consist of more lipase known as enteric lipase. Okay? So there's a pancreatic lipase, then you have enteric lipase, and the fat here you can see is converted into emulsified fat. In this emulsified fat is acted by the pancreatic lipase into fatty acid and two monoglycerides. In the diagram below you can easily now here the digestion in the different pH present in the whole digestive tract. You can see the saliva is at 6.5, which is almost a neutral kind of pH, in which there is composition of amylase bicarbonate, whereas gastric juice you have basically, so HCl is basically present, due to which the pH is 1.5. There's pepsinogen and renin in ruminant mammals. You have bile, in which is towards the basic side or alkaline side, which is around 7 to 8 pH, which is bile salt and pigments. Pancreatic juice is 7 to 8, again towards more like of your alkaline side, whereas membrane enzyme is again 7 8. Membrane enzymes are mostly present in the spleen. Finally, from the whole process of the food entering from your stomach to the till your small intestine, the digestion takes place. Then this digestion of the nutrients then further have to be absorbed inside the body, right? So to absorb these nutrients we have different processes or transport through which these nutrients enter into the body. Now these are through passive diffusion, facilitated diffusion, active transport and endocytosis. The passive diffusion is basically, we know that diffusion is from a higher concentration to a lower concentration, there is no energy required, whereas facilitated diffusion is from higher to lower concentration, but there is a need of some sort of a carrier protein here, though no energy is still required in facilitated diffusion. But in active transport you have energy required, and it is from a lower concentration to a higher concentration. So here at active transport you have the movement from lower to higher concentration. And endocytosis, cells form a vesicle to surround and engulf. You know, endo means to take in, cytosis means cell, so cells takes in the vesicle itself or engulfed. Now absorption, you can see how we have, there is a diagrammatic presentation. The passive diffusion occurring from the higher to the lower concentration. Then you have facilitated diffusion. You can see that there is a carrier protein present which is responsible for the diffusion. Now here in the active transport you can see there is a carrier protein, there is use of energy, and but there is the diffusion is, or the transport, sorry, is from lower concentration to the higher concentration. And finally your endocytosis, in which the nutrients are engulfed by the help of the membranes, cell membranes engulfs the absorption. We have been studying a lot about the villi. You can see here that these villi then consist of an artery, there are veins, there is the artery carries ends at a point, and the food is then being, or the nutrients are then being absorbed into the bloodstream. In figure B you can see a cross-sectional diagram of how brush borders are there, the basement membranes, and how the central lacteal, which is the lymphatic system again, and the arteries and the veins, the capillaries are present for the absorption of the nutrients through the different processes I have already discussed with you. Now see absorption of carbohydrates, how the carbohydrates are being absorbed from the intestinal lumen into the blood. The microvilli are present. Now glucose enters into the cell, epithelial cell, through sodium glucose symporter. See, symporter means there is some protein or present which is again with sodium, the glucose both enters into the cell driven by high extracellular sodium. Now they then from the epithelial cell further enters into the blood through glucose uniporter, which is GLUT2, which facilitates the downhill efflux. Okay, so this is how 2 potassium, 3 sodium, as the sodium potassium ATPase pump is present, and the glucose uniporter is present for the transport of glucose. Now here you can, this is another diagrammatic presentation of how the GLT1 then enters, causes the entrance from the intestine into the epithelial cells of glucose, and this through GLUT2 then finally enters into the blood. Similarly, the fructose enters into the epithelial cells through GLUT5, which is a uniporter protein, and then it finally enters through GLUT2 into the blood. Absorption of amino acids is through the proteins digested into oligopeptides and amino acids, then enters through the brush borders. You can see they can enter into the through the oligopeptide को ट्रांसपोर्ट, ठीक है, यहाँ पे हाइड्रोजन भी साथ एंटर हो रहा है पेप्टाइडेज के थ्रू, और इसी तरह जो है सोडियम जो है वो भी एंटर हो रहा है अमाइनो एसिड को लेकर, ठीक है. सो देयर इज़ अमाइनो एसिड को ट्रांसपोर्टर्स, देयर इज़ ओलिगोपेप्टाइड को ट्रांसपोर्टर, ठीक है, and the brush borders which transfers from the lumen into the enterocytes, and finally to the amino acid transporter, the amino acid from the enterocytes enter into the bloodstream. You can easily see below there is a bloodstream underneath the enterocyte. Now this is how it is presented for the fatty acids. Okay, fatty acid, obviously fats, the triglycerides into fatty acids from the pancreas. They are then miscellaneous or solubilization with bile acid, and this then the jejunum causes the esterification. The fatty acids then divide into triglycerides, and triglycerides into cholesterol, phospholipid, beta lipoprotein और chylomicron formation होती है, and this finally enters to the tissues for utilization of fat, and these fats see are transported through your lymphatics. Now here it is a representation how the ApoE see how they can transfer the now from this is whole schematic presentation of how from the food enters into the mouth, esophagus, stomach, then the secretion from the pancreas and liver, the lipids enter into the lymphatic system and then the bloodstream, whereas absorbed food, absorbed water enter into the hepatic portal vein, and this vein then goes to the heart. And unabsorbed food or undigested food are finally excreted into the anus. Nutrients are absorbed into the circulatory or the lymphatic system. They are water soluble nutrients which easily enter into the circulatory system, okay, like carbohydrates, amino acid and water soluble vitamins. To the GI tract capillaries, the water soluble nutrients to hepatic portal vein and water soluble nutrients in the liver. So this is how the whole absorption process takes place, and the liver is the whole transport of the blood taking basically from the small intestine and from the stomach are then entering this whole blood from here enters into the stomach, from the stomach and the small intestine enter into the liver, and in the liver this food is further detoxified. The detoxification occurs. Any toxic substances present in these nutrients are neutralized. Liver has one major action to neutralize the toxic substances. Now coming to the malabsorptive syndromes, we have the defective nutrient absorption. Basically we have studied absorption and digestion here ना. So मैल एब्जॉर्प्शन में क्या है कि या तो डाइजेशन सही नहीं हो रही या फिर कोई एब्जॉर्प्शन का इशू आ रहा है, विच कुड बी ड्यू टू एनी म्यूकोजल डैमेज, enzyme deficiency or motility. Now celiac disease, you can see here, is an autoimmune disorder which is triggered by gluten. Gluten is basically present in wheat, in barley, and this gluten could trigger a celiac disease which affects in a bilious atrophy. The villi here, see a typical gut in the picture, and the villi in the celiac disease, so they are flattened, which causes malabsorption, and when there will be no proper absorption, that undigested food will be excreted out from the body through diarrhea. Now do you have tropical spruce? Tropical spruce is basically very common in the tropical regions, like here it is mentioned Caribbean, Southeast Asia, India, and here it is caused by chronic infections, right? Chronic infections की वजह से हो रहा होता है, and this is so features chronic diarrhea, nutritional deficiency, folate and B12 deficiency. As I have discussed, like if there is no intrinsic factor, there will be no vitamin B12 absorption, and when there will be no vitamin B12 absorption, there will be anemia, and that anemia is called as pernicious anemia. ठीक है, तो इसमें भी यू हैव फ्लैटेंड विलाई. ठीक है, फ्लैटेंड विलाई होंगे, बट द कॉज इज़ अननोन. कॉमनली कहा जाता है कि बैक्टीरियल इन्फेक्शन की वजह से, रिकरेंट बैक्टीरियल इन्फेक्शन की वजह से हो रहा होता है. देन यू हैव IBS or irritable bowel disease. Now there is no functional GI disorder, and irritable bowel disease is caused by multifactorial things, which could be stress, which could be infection, but there is no major common थिंक प्रेजेंट कि हाँ इसी वजह से हो रहा है. देयर इज़ नो स्ट्रक्चरल इंटेस्टाइनल डैमेज बट द सिम्टम्स आर एब्डोमिनल पेन, altered bowel habits or bloating. I hope you have understand the whole process of absorption and digestion. If you have any queries or questions regarding this presentation, you can feel free to ask me or come to me whenever you are coming to the college, and we will make it clear. Okay, thank you. Allah Hafiz.